Good morning, everyone. Thank you for joining us this morning. And though our Heart Month is officially over as of last week, we have a bonus vascular-related talk this morning that I'm really excited about. Dr. Elaine Shelton is joining us from Nashville, Tennessee, where she's an assistant professor of pediatrics and pharmacology at Vanderbilt University Medical Center. Dr. Shelton completed her PhD at Cincinnati Children's Hospital Medical Center, where she focused on defining transcriptional regulators of heart valve development. And then she moved to Vanderbilt for a postdoc position in cardiovascular medicine. And since her postdoc position, she was focused on understanding blood vessel development, repair, remodeling. And she's continued that work. She was recruited to the Division of Neonatology at Vanderbilt, and she works on mechanisms of fetal blood vessel development and cardiovascular transition at birth. She's particularly interested in understanding the molecular and genetic regulation of ductus arteriosus, which is an essential fetal artery, and looking at to develop novel therapeutics to treat patent ductus arteriosus. So I think her work is really nice. It's very interdisciplinary. So she uses transcriptome analyses, high throughput screening, but also things that would be maybe of interest to engineers in the crowd. So vessel myography assays, and really looking at the interplay between molecular and biomechanical factors that regulate the ductus development. So I'm really excited to have her here today. And Dr. Shelton, is all yours.
Hi, thank you so much for that nice introduction. I'm gonna share my screen. Okay, hopefully everyone can see this. Thank you so much for inviting me to talk today. I just want to start by giving everyone a little background information on the ductus arteriosus. So this is a really essential fetal artery that's really, it's essential in utero. It connects the main pulmonary artery to the aorta, and its role during development is to shunt blood away from the developing lungs and put it back into the systemic circulation so that that blood can be oxygenated by the placenta. However, soon after birth, as soon as the infant's born and takes their first breath, their lungs inflate, that shunt vessel needs to rapidly close and seal itself off forever so that proper perfusion of the neonatal lungs can be achieved. There are really two basic phases of ductus closure. So over here on the left, these pictures are of a mouse ductus and a mouse heart, but the same thing happens in humans. So the first phase is called functional closure, and this is basically just the vessel constricts and occludes itself. And so just to orient you, so that this is the heart here, and it's kind of tilted sideways, and here's the ascending aorta, descending aorta. This is the main pulmonary artery. Here's a branch pulmonary artery going to the lung. There's another branch artery that's kind of going into the picture that you can't see. And right here is the ductus. And so it connects the main pulmonary artery and the aorta. And these hearts have been injected with a blue ink just to help us visualize the vasculature. So you can see here at birth, the ductus is wide open. There's a lot of blue ink in the lumen. But by 30 minutes to an hour after birth, you can see that the lumen is narrowing and the walls are thickening of the ductus here. And by 4 hours after birth, that lumen is totally occluded and functional closure has been achieved. So in mice, it takes about 4 hours. In humans, it takes anywhere between, in a normal term human birth, it takes anywhere between 12 to 48 hours. The next phase of ductus closure is the anatomical remodeling stage. And here, this is a human heart, and here's the ductus, or what used to be the ductus. And in this anatomical remodeling stage, that blood vessel has completely transformed itself into a structure called the ligamentum arteriosum. So it's no longer a blood vessel. It doesn't have smooth muscle or endothelial cells anymore. All those cells have either died or transformed into fibrous connective tissue. There are a couple main factors that keep the vessel open in utero. So things like low oxygen tension, the fetal environment is hypoxic compared to the newborn environment. Nitric oxide and prostaglandin signaling, those are both vasodilators. Those are very important in maintaining vessel patency in utero. And then just generalized vessel immaturity. So the smooth muscle cells aren't quite developed yet. A lot of the sensing, the oxygen sensing and other signaling mechanisms haven't fully come online yet. So those are all things that keep the vessel relaxed in utero. However, right after birth, we know that a sharp spike in oxygen tension as soon as the infant takes its first breath. Oxygen signaling is a really strong trigger for initiating smooth muscle contraction in the ductus. And then you have a sharp decrease in dilating factors like prostaglandins. And then just generalized vessel maturity. The smooth muscle is mature and can facilitate a strong contraction that would help to seal the vessel off. And then things like antenatal steroids. So things like betamethasone, if a doctor thinks that a mom's going to go into preterm labor, they often give antenatal steroids to just help mature many organ systems, the lungs primarily, the skin, but also those antenatal steroids have a positive maturation effect on the cardiovascular system and specifically the ductus. So that's what happens normally, but what happens when good vessels go bad? So here in this diagram, just to orient you again, this section right here is the ductus, and this is showing what happens when the ductus remains open after birth. That's a condition called patent ductus arteriosus, or PDA. So PDA is a persistent left to right shunt. And you can see that instead of all of this oxygenated blood going out to the systemic circulation, because this shunt is still open, some of that blood recirculates to the lungs. And so that causes overcirculation of the lungs and a decreased perfusion of the periphery. That can lead to things like pulmonary hemorrhage and respiratory distress syndrome, things like intestinal perforation and neurodevelopmental impairment. So this is a really serious problem that accounts for approximately 10% of all the congenital heart defects that are reported, and it disproportionately affects preterm infants. So 60 to 80% of infants that are less than 1,000 grams are afflicted with a vessel, a ductus that doesn't close. Okay. We know that there are several risk factors that are associated with PDA. So things like early gestational age, low birth weight, sepsis. There are also drugs that that either mom takes or babies given in the NICU. So things like furosemide or antihistamines that that are associated with a higher incidence of PDA. Right now, the current therapeutics are pretty limited. There are basically 3 drugs that you can give an infant to encourage the ductus to close. Those would be indomethacin, ibuprofen, and acetaminophen. All 3 of those are non-selective COX inhibitors, which inhibit the prostaglandin or PGE2 pathway. They're not specific, so they don't specifically target the ductus, and so they can cause kind of generalized vasoconstriction. So the cerebral vasculature will constrict, the gut will constrict, the renal vasculature will constrict, which is not ideal for a preterm infant. And they're actually ineffective in approximately 30% of infants that require therapy. They also have some pretty severe side effects in some patients, like intestinal perforation that can be life-threatening. So if the vessel doesn't close on its own and the drug intervention is ineffective, infants have to go to the OR to get a surgical ligation of the vessel, because you need that vessel closed in order to properly perfuse your other organ systems. Again, that's kind of it does fix the problem, but it's also not ideal for fragile preterm neonates that often have other problems besides a ductus that won't close. There are cases in some congenital heart conditions that will require that the ductus remain open to keep the blood flow going. And so in those cases, those infants are infused with prostaglandins or stent implantations so that the ductus remains open until they can go and have the corrective surgery for their ductal dependent cardiac lesion. But again, these therapies are they're not specific, they're not ideal. They dilate all the vascular beds in the body, and they can lead to things like apnea, which again, for preterm neonate is not ideal. So the bottom line is that we need to develop more DA-specific therapeutic options that target pathways other than the prostaglandin pathway and that are safer and more effective than what's currently available. So that sounds like a nice goal, but it's not quite as easy as it may sound. So the success rate of drug development programs is actually staggeringly low. Only 4% of compounds that enter into clinical trials actually yield licensed drugs. But there have been studies that have characterized the features of clinically successful drug targets. And so, first of all, the first characteristic of a drug target that actually leads to a licensed drug is that it's enriched in the tissue of interest. And so in our case, we want things that are heavily expressed in the ductus compared to the surrounding vessels, because we want a drug that will specifically target the ductus and leave all the other vascular beds alone. We want this target to be part of what's known as the druggable genome. So this is a subset of about 4,500 proteins that are likely to bind drug-like molecules. So we want something that we can actually target with a drug. So these are things like enzymes or receptors or ion channels or transporters, things that are easily accessible to drugs. We also are looking for a drug target that actually has some association to either ductus development or PDA. So these would be things like genes that are involved in mouse models of PDA, or single gene syndromes that are associated with PDA, or we've identified some non-syndromic SNPs that are associated with PDA. And then we've also just experimentally shown that some genes or pathways are play a role in regulating DA development or contractility. So we've kind of come up with a short list of potential targets. And again, we're trying to look for something that lies in this middle region. So we want a potential drug target to be enriched in the ductus, part of the druggable genome, and linked to PDA. So these are just kind of a subset. This isn't an exhaustive list, but these are some of the candidates that we've come up with that actually fulfill all three of those requirements. So you can see there's a lot of ion channels on there, there's enzymes, there's G protein coupled receptors. And so today I'm going to tell you about a story that involves these two genes in particular, ABCC9 and KCNJ8. And these two genes make proteins that form the two subunits that comprise ATP regulated potassium channels, or KATP channels. And this is what a KATP channel looks like. So the inner region of the channel that's formed by what are called KIR or KIR subunits, those are coded for by KCNJ8. And they form the pore region. And then ABCC9 is a gene that codes for the SIR subunit, which lies along the outside of the channel. And that's kind of the regulatory region of the ion channel. So ion channels are great drug targets. They're very accessible. And for KATP channels in particular, there are a lot of different isoforms of KATP channels. And the great part about that is that they're tissue specific. So KIR 6.1 and SIR2B, that combination of subunits is expressed in the vasculature. There are other isoforms like the 6.2 SIR2A combination that is expressed in the heart, and then you have the 6.2 SIR1 combination that is expressed in the pancreas and in the brain. So this is kind of an ideal situation for us because we have something that we know is enriched in the ductus, and it's a target that is this is something that we can easily target because it's an ion channel, and it's kind of sequestered. This subunit combination is sequestered to the vasculature, so we can target this combination of the subunits without disturbing the KATP channels that are in the heart or in the pancreas or in the brain. So we also know that KATP channels have a relationship to ductus development and function. So human patients that have an autosomal dominant disorder, which is a gain of function mutation in either subunit, either KCNJ8 or ABCC9, have what's called CAN2 syndrome. And so these are pictures of patients that have CAN2 syndrome. It's a rare disease. It actually has a host of different manifestations. So some of them are cardiovascular, some of them are neurodevelopmental. But the one that obviously sticks out to me is that 50% of CAN2 patients have a significant PDA that is resistant to drug therapy and often has to undergo surgical ligation. So we know that KATP channels play some role, or it seems they play some role in either the development or the function of the ductus. And I'll keep coming back to this, but people with CAN2 syndrome have a gain of function mutation. So that means that they have overactive or constitutively active KATP channels. So some of the work that we've done, first we wanted to confirm that KATP channels are indeed enriched in the ductus. And so to begin to do that, KATP channels have come up in our different transcriptome analyses. So we've done microarray analyses of mouse ductuses. We've done RNA-seq analyses of human ductuses, and KATP channels, those two subunits, have popped up on both of those transcriptome analyses. But we just wanted to confirm that the protein also was expressed at higher levels. And so to do that, we did antibody staining for KCNJ8 and ABCC9. So these are sections through the outflow tract of a mouse. And then we stained with antibodies specific to KCNJ8 or ABCC9. And so here's the main pulmonary artery, here's a branch pulmonary artery, and here's the ductus region. And in both cases, you can see that both of these subunits, the proteins for these subunits, are highly expressed in the ductus, and they're expressed at lower levels in the surrounding vasculature, which is good news for us. We also wanted to see what the expression pattern is in human samples. So we acquired some human ductus samples from our colleagues in the surgical center. And this is, I know this might be a little bit hard to see, but this is a cross section through a human ductus. Here in the white space, this is the lumen of the blood vessel. This is what's referred to as the intimal cushion region. And then this would be the contractile smooth muscle region. And so in this experiment, we did an RNA scope analyses, which basically localizes transcripts for genes. And so these purple dots here that you can see in this inset represent individual transcripts of ABCC9 and KCNJ8. And so the interesting thing about this is that both genes seem to really be heavily concentrated in the contractile smooth muscle region of the vessel, as opposed to the cushion region or the endothelium here. So this really seems to be a smooth muscle specific expression in human ductuses, at least. So with those two experiments and our transcriptome analyses, we feel pretty confident that the KATP channels are enriched in the ductus. Okay, So on to our functional studies. We use a technique called cannulated vessel myography, where we dissect out fetal ductuses from mice, and we mount them in microvessel perfusion chambers, which you can see here. So that's what this looks like. So we mount them on some glass pipette tips, and they're in an organ bath system. There's a buffer, just a saline buffered solution that's circulating around the vessel, and we can spike drugs in and out of the perfusion chamber. And then we have software that measures the lumen diameter in real time. And so we can record and quantify the effects of different drugs and whether that's causing vasoconstriction or vasodilation. So in this movie, we've spiked in potassium chloride, which is a very strong vasoconstrictor. And so you can see that when we spike in potassium chloride, the vessel constricts. And again, we have software that measures the changes in lumen diameter, and we can quantify that. So this is what a typical tracing, a typical myography tracing looks like. So in the beginning of each experiment, we inflate our vessel to normal fetal mouse blood pressure, which is 20 millimeters of mercury. And then we spike in potassium chloride just to make sure that we have a good, viable, reactive vessel. Then we can wash that out. So lumen diameter is over here on this side, and then time is on the x-axis. So a decrease or a contraction is illustrated here. And then when we wash it off, the vessel goes back to normal or resting lumen diameter. And then in this experiment, we have added increasing concentrations of a KATP channel activator called pinacetyl. And so each of these arrows indicates where we added the increasing dose of the drug. And you can see that pinacetyl causes an increase in lumen diameter, which to us suggests that activating KATP channels causes ductus dilation. And that's something that we've seen, that relates well to the Cantu syndrome phenotype, where they have constitutively active channels and they have vessels that remain open after birth. And so here we've done this experiment many times and in 3 different mouse models. So our wild type, as you add increasing doses of pinacetyl, the vessel dilates. We have a mouse model of a KR 6.2, which is the isoform that's in the heart. If you knock that out, the ductus doesn't care. You can add pinacetyl and still get a dilation, because 6.2 is not the isoform that's specific to the vasculature. But if you knock out KR 6.1, which is the vascular-specific isoform, you have very little reactivity to pinacetyl. You do get a little bump up here with the highest doses, but we think that that's just a non-specific potassium channel opener effect. So this experiment to us, we concluded from this that 6.1 is the important isoform in the ductus, and that if you activate KATP channels in the ductus, that causes vasodilation. We then did kind of the converse experiment, where we looked at increasing concentrations of KATP channel inhibitors to see if we could induce vasocontraction, and we did. But we didn't just do it in the ductus. We also mounted ascending and descending aortas, as well as mesenteric arteries, because again, if we're thinking about drug discovery, we want something that specifically targets the ductus and leaves the other vasculature alone. We had a hunch that KATP channels are enriched in the ductus, and so we were hoping that even if the aorta and the mesenteric arteries had some baseline level of expression, we would be able to find a dose of an inhibitor that would enact an effect on the ductus, but would not have an effect on the other vasculature, because the expression of KATP channels would be so low in those structures that the inhibitor wouldn't have much of an effect. And really that's what we saw. I know these are a little bit of confusing graphs here, but glibenclamide is an inhibitor that non-specifically targets the SIR outer regulatory subunit of the channel, and this compound PNU 37883 is a non-specific KIR subunit inhibitor. So when you dose the vessel with increasing concentrations, you can see that you get a decrease in lumen diameter, and that happens not only in term ductus vessels, but also in very preterm fetal ductus vessels as well. Whereas the aorta and the ascending and the descending aorta, as well as the mesenteric artery, are relatively unaffected by inhibiting channels. We then wanted to move on to some in vivo studies. So in these studies, we delivered mouse pups, term gestation mouse pups by C-section. And again, just to remind you, at birth, the ductus in a mouse should be wide open, which is what you see here. But by 4 hours after birth, it should be closed, which is what's illustrated here. But in neonates that we injected with pinacidil, which is the KATP channel activator, when we looked at their ductuses 4 hours after birth, we saw that pinacidil had the effect where those vessels remained open at the time point when they should have been closed. So this might be similar to the CAN2 situation again, where you activate the channels and you don't get the ductus closure that you would expect to happen. Here's the converse experiment where we were trying to look at the effects of KATP channel inhibitors in vivo. And in this experiment, we didn't inject pups this time. We injected the pregnant dams with either glibenclamide or that PNU compound 1 hour prior to delivery. And then we looked at the state of the pups' ductuses immediately at birth. So in a vehicle control situation, as expected, the vessel is wide open at birth. But in both cases, if you inject the pregnant mom mouse with either glibenclamide or PNU, which crosses the placenta and is able to affect, is able to infiltrate the pup, you can see that at birth, in both of these situations, you have a narrowing of the ductus, thickening of the walls, and a narrowing of the lumen here, which suggests that in vivo, if you inhibit KATP channels, you cause vasoconstriction here. Okay, so I mentioned earlier that oxygen signaling is a main regulator of ductus contractility, and in preterm infants, they have less oxygen sensing than term gestation infants. So they're less sensitive to oxygen than their term gestation counterparts, and preterm mouse pups behave in the same way. So in this experiment, we took day 15 of gestation. So in a mouse, 19 days is considered full term. So if we take the pups out at 15 days of gestation, that's equivalent to a preterm human infant. If we expose that vessel to 12% oxygen, we get a modest constriction. Not as much as KCL, that very potent vasoconstrictor, but we get a modest constriction. But if you pre-treat that same vessel with PNU or glibenclamide, something that would cause the KATP channels to close, and then you expose that vessel to oxygen in the context of inhibited KATP channels, you get a much more robust constriction, suggesting that if you close KATP channels or if you inhibit KATP channels, you can make a preterm ductus more sensitive to oxygen. And we did that same experiment a slightly different way. We took a genetic approach this time where we used preterm wild type vessels or preterm vessels that had KR 6.1 knocked out, so they don't have functional vascular KATP channels. And we exposed both of those vessels to a series of increasing concentrations of oxygen. And so in the wild type vessel, you see that you do get constriction with each increasing percentage of oxygen. But in the preterm vessel, you get a much more robust constriction at each stage, suggesting, again, that in a situation where you don't have functional KATP channels or you have inhibited KATP channels, those vessels are more sensitive to oxygen-induced constriction. Okay, one more set of experiments in the context of oxygen. So if you deliver pups, if you deliver pups in room air by C-section and look at their ductuses 4 hours after birth, you see a totally closed vessel. This is the normal situation. If you put pups into a hypoxic chamber where you lower the concentration of oxygen to 12% instead of the normal 21%, which is what's in room air, if you deliver pups and put them in a hypoxic chamber for 4 hours and then look at their ductuses, because they're not getting as strong of an oxygen stimulant, their vessels remain open 4 hours after birth. And so we hypothesized that perhaps if you inhibit KATP channels and then expose newborn mouse pups to a hypoxic environment, could inhibiting KATP channels improve ductus closure in that situation. And so that's what we did. We injected newborn mouse pups with glibenclamide, which is the KATP channel inhibitor, 30 minutes after they were born. And then we put them in the hypoxic chamber for 4 hours. And you can see, it's a little bit hard to see, but hopefully you can make it out, that this vessel, there's still maybe a tiny trickle of blood that's going through that lumen. I can see just a little strand of red there, but it's definitely more closed than in the uninjected or the vehicle injected control pups at 4 hours. So that suggests to us that glibenclamide inhibiting KATP channels can induce ductus closure even in hypoxic situations. So all of that work was done in mice, and figuring out PDA in mice is a great goal, but ultimately we want to figure out how to solve PDA in humans. So we're trying to now do studies using human vessels and human cell lines. So I won't go into all of the detail here, but basically cell lines for human ductus, endothelial and smooth muscle cell types have not previously been available. So we took on the challenge of trying to make some immortalized cell lines using cultured human ductus. We get human ductus pieces from the OR, and then we can grow those cultures out, and then we can immortalize them using the SV40 antigen immortalization technique. And so we were able to get some smooth muscle cultures as well as some endothelial cultures that we confirmed with antibody staining and lots of PCR. And then we did a RNA-seq experiment just to make sure that our cell lines that we created retained their ductus identity. So we just didn't want to make generic smooth muscle or generic endothelial cell lines. So we did an RNA-seq experiment where we took all of our cell lines and we compared the endothelial ones to HUVECs, and they are distinct populations. And then we compared our smooth muscle cell lines to either neonatal aortic smooth muscle cells or adult aortic smooth muscle cells. And again, our ductus smooth muscle cell lines were a distinct cell population. So we're pretty convinced that the cell lines that we made are actually ductus specific endothelial and smooth muscle cells. So using those cells in this experiment, this is just smooth muscle cells in these cultures, but we grew aortic smooth muscle cells and ductus smooth muscle cells either in room air, regular cell culture incubation conditions, or in a hypoxic incubator that we lowered the oxygen content down to 2% oxygen. And here, this is just a quick experiment, qPCR experiment. So we already know that, that ductus smooth muscle cells have a higher expression of KCNJ8 and ABCC9, the two KATP channel subunits, compared to aortic smooth muscle. So that was not a surprise to us. But the interesting result here is that if you compare the two ductus bars, putting ductus smooth muscle cells into hypoxic conditions increases the expression of both subunits. So suggesting that hypoxic conditions promote KATP channel expression. which is interesting because in utero, you're in a hypoxic environment and you also want the vessel to stay open. We know that KATP channels promote vasodilation. And so it makes sense that in hypoxic conditions, you would want to promote the expression of a factor that would maintain that vessel patency in utero. Another experiment that we do with the human vessel samples that we get from the OR is we do tension studies. And so this is like a myography experiment, but because these vessels are so large compared to the fetal mouse vessels, we have to do tension studies instead. And so in this situation, we string up the human vessel. We connect it to 2 wire hooks, one of which is connected to a stationary glass piece in this organ bath. So this stays in place. The other glass hook is connected to a force transducer that gives us a readout of tension. And so if you spike in a drug that causes vasorelaxation, you'll see a reduction in grams of tension. And if you spike in something that causes the vessel to constrict, you'll see an increase in the grams of tension that is read out by the transducer. So here's a tracing of these studies, and actually you read them in the opposite way that you would the first myography tracings that I showed you. So in this case, a dilation is actually an increase. A constriction. A constriction is indicated by an increase in tension. So if you see, if you go up on the y-axis, that's actually a constriction, and a dilation is down on the y-axis, which is a little bit different than the mouse studies that I showed. But in any case, this is just one example tracing of a study we did. We found that if we spike in this compound U46619, which is a known vasoconstrictor, it's a thromboxane receptor agonist that we know constricts the ductus, we get a nice constriction of our human vessel. And that can be overcome when we spike in panacetyl, which is the KATP channel activator. So you get a nice dilation. If you wash that off and you change the gas conditions that you're bubbling in the organ bath, if you start bubbling in 21% oxygen, you get a slight contraction. But if you then add glibenclamide on top of that, again, it seems like inhibiting KATP channels makes human vessels even more sensitive to oxygen as well. So the next step of this project is now, the data that I've been showing you, panacetyl and glibenclamide are pretty non-specific activators and inhibitors of KATP channels. So we really want something that's only going to hit the vascular isoforms of these channels, because we don't want to have any off-target effects of KATP channels in the brain or in the pancreas or in the heart. We really just want to target the vascular channels. And up until now, specific vascular KATP channel drugs have not been available. So working with medicinal chemists and people in pharmacology at Vanderbilt, we're now running studies to help to identify vascular-specific inhibitors of KATP channels. And I won't go into all the nitty-gritties, details of this high throughput screen, but basically we use this thallium flux fluorescent assay. And so if you have a cell that fluoresces, that basically means that you're getting an activation of KATP channels. So here, if you spike in panacetyl, which is what you see with the blue line here, you get an activation or an increase in fluorescence, suggesting that you've activated a KATP channel in a specific well of the plate here. And if you treat with panacetyl and, in this case, glibenclamide, you get a decrease curve from that initial panacetyl curve. So this is just for reference. We use glibenclamide just kind of as a as a control because we know that it closes KATP channels. But then, in every other well of this plate, we spike in, different isoforms of our small molecules, just to try to find ones that will actually inhibit the KATP, the vascular KATP channels. So we've identified two new compounds. And so once we get compounds that we think look interesting, then we go back to our mouse myography studies to see if they do have an effect on vessel contractility. And so in this case, the blue line represents glibenclamide, and I've already shown you some of this data that suggests that increasing concentrations of glibenclamide cause vasoconstriction. And here are two new compounds. So this VU, Vanderbilt University 063, this we think hits the 6.2 SIR1 combination, which is not the vascular specific combination. So when we put increasing concentrations of that on the vessel, we see no change, which is great. But here's kind of the exciting data is this compound, VU278, we think is specific for the vascular channels 6.1 and SIR2 combination. And when you put increasing concentrations of that on the vessel, we do see a nice vessel contraction, which is similar to what we see with glibenclamide. So this is a compound now that we're working up with more detail and moving to kind of our whole animal mouse models to see if this induces contraction in those systems as well. So just to kind of summarize what I've told you today, KATP channels, we know, so this is a poor representation, but it's supposed to be a smooth muscle cell. And we know that KATP channels are enriched in the ductus, smooth muscle. If you activate KATP channels using something like panacetyl, you actually induce membrane hyperpolarization, which decreases intracellular calcium accumulation, leading to vasorelaxation. I didn't talk about these steps here, but we've measured membrane excitability and calcium levels in our cells. And so we know that this is the pathway that's going on in our smooth muscle cells. And so you get muscle relaxation, which equates to vasodilation. And we've seen that in delayed ductus closure in our in vivo mouse models. And that's also what's probably happening in Cantu syndrome and in patients with Cantu syndrome. But if you block or if you inhibit KATP channels, this doesn't happen. And so you get an accumulation of calcium, which leads to vasoconstriction instead. I didn't talk much about this, but oxygen is, we know, is a big trigger of vasoconstriction through several different pathways, but it all culminates in increasing calcium accumulation, which then leads to smooth muscle cell contraction. And so if you inhibit, so that we know that there's a relationship between oxygen and KATP channels. We know that KATP channels can, if you inhibit KATP channels, you can make a preterm ductus more sensitive to oxygen. And our cell culture experiments where we put human ductus smooth muscle cells in hypoxic conditions, we know that hypoxia can upregulate the expression of KATP channels. So all that is to say that we think that vascular-specific KATP channel inhibitors may be an effective alternative therapeutic for treating PDA, particularly in those premature neonates that are not very sensitive to oxygen and really require an increase in oxygen sensitivity, number one, to help their natural vasoconstriction signaling pathways become more robust, but also a therapy that doesn't affect things like the renal vasculature, the cerebral vasculature, the gut vasculature. So we think that KATP channel inhibitors that are highly enriched in, KATP channels that are highly enriched in the ductus, if you can inhibit those with vascular specific compounds, that might make for an effective therapy for PDA. And with that, I will, I think I'll stop there just so we have some time for questions. I haven't looked at the chat. I don't know if we have questions in the chat or not, but if we do, I can stop and take questions. And if not, I have a couple other data slides, but it's up to you. If I can't see what's going on in the chat.
There's a few questions there,
Okay.
but I mean we have a few minutes. So I don't know if you have something you would like to show. It's
oh well, let's do some questions and
okay.
then we'll see where we are.
All right. So these questions I think all come from Daniel Chaparral, who's a PhD student in my lab and had noted offline how impressed he was with the myography setups and stuff too. So I think he's looking forward to talking with you about that later. He asked, do the results of the combination of oxygen concentration with the KATP inhibition remain specific to the DA?
Yes. So the ductus is uniquely sensitive to oxygen. It's the only vessel that constricts at birth in response to oxygen. So while we've put just oxygen on other vessels. So we've looked at the effect of increasing oxygen on the aorta, and the aorta doesn't care, which is good because you don't want the aorta to constrict in response to oxygen. So we don't have any reason to believe that using glibenclamide in addition to increasing oxygen will affect, other vessels. We haven't specifically tried that, but because glibenclamide by itself isn't very effective on the other vessels, and oxygen by itself isn't effective on the other vessels, we don't think there's a problem there. But certainly that's something that we should test before we try to get a clinical trial started in infants.
So I'm going to piggyback on his then before I get to his other questions and ask. So kind of two things there. The ductus muscle cells, you show that they're distinct from muscle cells of other vasculature. So in this oxygen response, I guess is perhaps unique to them, like you're saying. So where does that difference in this muscle cell population arise? Is that developmentally, they come from different developmental populations?
Well, so that's a great question. So it's interesting because the smooth muscle of the ascending aorta and the smooth muscle of the ductus are both neural crest derived. So they have the same embryonic origin, but they have vastly different transcriptional profiles. So there's something besides embryonic origin that contributes to those transcriptional differences. We think that some of it is probably epigenetic, although we haven't explored that in depth. But some of it we think is also probably hemodynamically regulated. So there's in utero, there's not a lot of blood flow that's going through the ascending aorta until right after birth. And so the ductus is getting a tremendous amount of hemodynamic flow during development that the ascending aorta is not seeing. So we think that there's probably some hemodynamic, biomechanical, gene regulation programs that are happening in the ductus that are not happening in the ascending aorta. And just a big difference between the two vessels. The ductus is a muscular artery, the aorta is an elastic artery. So just on a very basic structural level, those vessels are just very different. And it's because they have different jobs to do. So
interesting. Cool. So Daniel also asks, is the increase of expression in the KATP channels due to hypoxic conditions in the in vitro experiments reversible?
So that's a great question. We haven't tried to reverse the phenotype. We just put them in hypoxia and then looked at gene expression. But that's a great experiment, because that's what happens, kind of in the in vivo situation. You move from a hypoxic environment where you would potentially have high expression of KATP channels, and then all of a sudden you're in a room air environment where you want to downregulate your vasodilators. So my hunch is that yes, it is reversible, but we haven't shown it.
And then this is framed from an optimistic PhD student. It says, this is amazing data and tells a compelling story. What's the ballpark timeline of all of this, I guess, in terms of clinical translation?
Great. I don't know. I mean, 10 to 20 years probably before it's actually a commonly used therapeutic. And that's if we're lucky and, are one of those 4% of drugs that are, that make it through all the clinical trials. Yeah, and so I don't know what the timeline realistically would be. And it's particularly difficult to do something like that because it's a vulnerable population. We're looking at, preterm neonates, which are not easy to get a clinical trial approved for. But glibenclamide, which is a non-specific, it's not an ideal choice, but glibenclamide has been used clinically in the NICU. And so I think if we pursue a clinical trial option sooner rather than later, we'll probably go the glibenclamide route because it's already being used in the NICU. It's not very prevalent, but it is used to treat hypoglycemia. It hits the KATP channels in the pancreas. And so, we might be able to figure out a dose that wouldn't affect insulin production, but might have an effect on helping to close the ductus. And that just might be an easier route to start with because it's already an approved, an FDA approved drug that's being used clinically. But, it's not certainly we want a vascular specific small molecule, but we're not there yet. So.
And then in terms of when PDA is usually, so when is it usually detected and when would that therapy be given? So how would it actually look clinically?
Yeah, so there's a lot of variation between NICUs across the country. So there are some places that give prophylactic indomethacin or ibuprofen. So as soon as the infant's born, if it's less than 1,000 grams and maybe less than 28 weeks gestational age, they start a course of indomethacin right away. But there's other institutions that, because indomethacin causes things like intestinal perforation, they don't want to expose every infant to that drug right off the bat. So they'll wait a week and they'll see if the ductus, has any reactivity on its own within that week. And if it's still a hugely, wide open pipe at 7 days of age, then they'll start, a 2-day course of indomethacin. But there's a lot of practice variation. And, this PDA is a real conundrum for neonatologists. It's just a tricky thing to try to manage. And some vessels will spontaneously close, maybe not in that 12 to 48 hour window like we like to see for term gestation infants. It might take a month, it might take 2 months, but they'll close spontaneously without therapy. But then there are other vessels that won't close on their own. And those vessels, the longer that they remain patent, there's been studies to show that the longer a vessel remains open, the higher probability you'll have of death in the NICU. So it's something that probably needs to be taken care of, but how they do it and when they do it, is variable across the country.
So a question from, so there's several that are pouring in now. A question from Dr. Sharad Ramaswamy. He says hypoxic environments have been shown to release cardiac regenerative factors to mitigate tissue damage. Have you observed any kind of signaling that suggests there's any kind of regenerative processes going on in the hypoxic environments?
Regenerative. No, but we're not looking for those. We haven't looked. We have kind of a subset of genes that are, the common players in our pathways, and we kind of do PCR panels based on those, known players. So we haven't looked at regenerative potential or regenerative factors. But if you have any, we've got lots of samples in the freezer. If you have any things that you want us to look at, we could probably do that pretty easily.
A question from Jay Young asked what the ductus is composed of, and are there similar arteries where this may cause an adverse reaction? So you looked at the aorta, but which is, so you mentioned is different, but are there similar arteries that might, have some off-target effects?
Yeah. So, well, the ductus is, like most arteries, it's 3 layers. So there's an adventitia, there's a medial muscle portion, and then there's an endothelial layer. So it's, kind of a basic structure. We're just now starting to do a single cell seq analyses to see if there are pockets of, smooth muscle and endothelial cell types that change either over the course of development or change in relationship to, other muscular arteries. I don't think it's very useful to compare an elastic artery to a muscular artery. Just, we know there are big differences there, but we're looking at other muscular arteries. We haven't, we've looked at mesenteric arteries because those are kind of a good surrogate for resistance arteries. We don't see high levels of expression in mesenteric arteries, and we don't see a lot of response to our activators or inhibitors when we spike in those drugs to mesenteric arteries. But no, we've done aorta, we've done mesenteric arteries, and those are the only two. So certainly there are lots of other ones out there, but just in vivo, it's the ductus seems like it's pretty unique. There's only, that's the only vessel that closes in response to oxygen in that perinatal period. So, yeah. So.
And then Dr. Brittany Gonzalez asked, she said she may have missed this, but is there a way to detect and predict that PDA will occur before the infant's born?
That's a good question. Well, so, we're starting to kind of look at that now. Vanderbilt is kind of they've gotten a leg up on this, where they have, for the last 20 years, they've been trying to collect patient genetic information and match that with the electronic health records. So now we're going back through with the help of mathematicians and bioinformaticists, and we're trying to look for trends either in mom, like what conditions mom has, whether that's diabetes or high blood pressure or medicines that mom takes, or things that happen to baby, either prenatally or in the perinatal period, to see if any of those things correspond with a higher incidence of PDA. We know things like if mom is taking SSRI antidepressants in the 3rd trimester, we know that that's actually associated with in utero ductus contraction. So they'll have a ductus that closes prematurely, and that causes overcirculation of the lungs in utero, which leads to pulmonary hypertension. But we haven't identified anything before birth in the infant that would suggest that they're more or less likely to have a ductus that doesn't close.
As well. So thank you again for joining us today, Dr. Shelton. And
Thank you.
thank you everyone else for joining as well.
Okay, thanks.